In the first video from this unit, we looked at nucleic acids and how nucleic acids are structured and how that structure enables their functions. The main function outside of heritability, or passing on information from generation to generation, that nucleic acids serve is in directing the production of proteins and the synthesis of proteins. And so this video is going to look at the first part of the process of protein synthesis, what we call transcription. In this video, we're going to look at how the information stored in DNA is transcribed into RNA, and then we're going to look at how eukaryotic organisms process their genetic information prior to the second step, which we call translation. In terms of biological information flow, let's take a moment and just make sure we understand where we are. We know that the information in DNA is going to be transcribed into RNA. And so looking at these molecules, we should be able to understand that the top molecule is DNA based on its structure. We see thymine, we see a double strand. And then the second molecule is RNA, which we can see based on the single strand and based on the fact that there is uracil replacing the thymine. One thing that you can notice is that the sequence of the RNA molecule is identical to one of the strands and opposite to the other strand. What we would say is that the strand that it's identical to is what we call the coding strand, and then the second strand in the molecule is what we call the template strand. When transcription happens, the enzyme that's going to transcribe the DNA is going to use the template strand. And so anywhere where there's a C, that enzyme is going to place a G. Anywhere where there's an A, that enzyme is going to place a U. And so what's going to happen is that you're going to wind up with a sequence that's identical to the sequence on the coding strand, except of course that thymine is being replaced by uracil. The second step in this process is translation. We're not going to talk about it particularly in this video, but we'll talk about it in the next video. We'll acknowledge that it's happening there. So transcription is going to occur in the nucleus of eukaryotes. Prokaryotes, of course, do not have a nucleus, so the region where the genome exists inside of prokaryotes is known as the nucleoid. And so we'll put that down, though I don't actually really care if you know it. And so this is a really important step in this process. If we didn't transcribe the information to RNA, we wouldn't be able to use that information in turn to produce a protein. So if we're looking at one gene somewhere on a chromosome, this diagram is just showing us how that gene is organized. We've got one strand of DNA. It's the template strand that we're looking at here. And so we can see a couple of different regions that are really, really important. The first is this region of the promoter. The promoter signals the start of the gene to the enzymes that are going to transcribe the gene. And so that enzyme is going to load itself onto the promoter of the gene, and then it is going to begin transcribing the region. And we can see that we transcribe this entire region in red. This is going to give rise to a molecule of RNA that is actually going to have the message of the gene. And since it has the message, we call it messenger RNA, or more typically we might identify it as mRNA. And then, of course, there's going to be a stop sequence that tells the enzyme where to stop transcribing the gene so that the transcript can move on to the next step in the process. This 5' and 3' UTR region you don't really need to worry about. It stands for untranslated region, which means that these sequences will be copied into the transcript, but once translation happens, it will not include those particular sequences. This diagram is showing us what this looks like in a little bit more detail. We've got the enzyme that is doing this work, which is called RNA polymerase, which is going to take the information in that template strand of DNA and use it to synthesize an RNA molecule. One thing to note is that there's a directionality involved here, just like there was in our discussion of replication in the first video. Nucleic acids can always only be made in a 5' to 3' direction. And you can see that in the strand. The direction of the synthesis is happening 5' to 3', and that template strand is being read in the 3' to 5' direction, because that's the only way that these things can work. So this RNA polymerase enzyme is the enzyme that does the work. It's the enzyme that uses the information in the template strand, the sequence of bases, A's and G's and C's and T's, and uses that in order to produce the new strand. And if you are looking at it more closely, you can also see that the sequence of that mRNA transcript is identical to the sequence of the other strand, the non-template strand as it's labeled here, or what I would call the coding strand. Transcription happens in 3 major steps. We've got initiation, elongation, and termination. In the initiation phase, we're going to have RNA polymerase attach itself to the promoter. And so what I've done here is just illustrated RNA polymerase as this kind of blue-orange circle blob. And we can see that it's arranged itself so that it's at the promoter. It'll then begin to transcribe the gene. And so that's the elongation phase. RNA polymerase will move along the gene And as it moves along the gene, it is going to produce an mRNA molecule, which I guess we can do in pink here, right? So we'll have this pink mRNA molecule that's being produced as RNA polymerase moves across the gene. We'll have our 5' end of the transcript at the beginning and our 3' end at the end. And then in the termination phase, when RNA polymerase reaches a stop signal, that mRNA is going to be released, and it's going to wind up moving on to the next phase of protein synthesis, translation, if it's going to become a protein. Or some transcripts just remain as RNA molecules and go off to play other roles in the process that we'll talk about in the next video. So those are the main steps of the process: initiation, elongation, and finally termination. The other thing that we'll look at in this video is how eukaryotes specifically process their mRNAs prior to sending them to the ribosome to be translated into proteins. We have the same diagram again, but with some additions so we can see what's happening. 2 things are going to be added to the transcript, and 1 thing is going to be removed. At the front of the transcript, we're going to put a modified nucleotide cap, which we call a 5' cap. And you can see it circled and highlighted there. It's a single nucleotide on the front of the transcript that signals to the cell this transcript is to be exported from the nucleus. The other thing that we're going to add is to the 3' end, or the tail of the mRNA, if you want to think about it that way. We're going to add a poly-A tail. That poly-A tail is just a chain of adenine RNA nucleotides. It could be hundreds or thousands of nucleotides in length. Once the transcript is exported from the nucleus, it is exposed to enzymes that will break down RNA molecules, what we call RNases. The RNases start at the 3' end of an RNA molecule and start chomping from that direction. And so by having a poly-A tail, you give the RNA a longevity in the cytoplasm of the cell so that it can interact with the ribosome and produce a protein. If it didn't have that poly-A tail, those RNases would start breaking down that mRNA immediately, and it would not be able to produce a protein. The other thing that's going to happen is you are going to remove segments of the mRNA itself, what we call introns. We can see the introns identified in this diagram as these gray segments in between these red segments, which are called exons. These introns have to be removed. They do not have any information that leads to the production of a protein. And so these are going to be removed by functional RNA molecules inside of the nucleus, and then the exons are going to be spliced together. So you can see that the mature mRNA is actually shorter in terms of its protein coding region than the transcribed region in the gene. And that's because these introns have been removed and these exons have been spliced together. All 3 of these modifications will occur prior to exporting the RNA from the eukaryotic nucleus, and they all need to occur if the goal is to produce a protein at the end. And so that's our discussion of transcription. As we wrap it up, make sure you can do the following things here at the end. Make sure you can describe the structure of a gene, including the promoter and the transcribed region of that gene. Make sure that you can explain the process of transcription, what happens in initiation, what happens in elongation, what happens in termination. And also make sure that you can describe how eukaryotic mRNA is processed and why those processing steps are required in the production of a functional mRNA transcript inside of a eukaryotic cell. If you can do all those things, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions that you have so that you can get the help that you need. Thanks again for watching. I really appreciate it. Have a great day.